Okay, so let's try an overview in a relatively brief fashion, the whole idea of the adaptive responses. And we're going to start with humoral adaptive responses. So the common feature of all adaptive immunity in humans is that it is rather slow. It requires days, if not weeks, to develop. It's extremely specific. Responses are targeting very particular pathogens. It is systemic, unlike the innate responses, which are more local. It's very efficient because it's so specific, which, if you compare with the limited efficacy or efficiency of the innate responses. And one of the fundamental features that innate responses lack and adaptive responses have is the feature of immunological memory. The ability of your immune system to mount more robust immune response and mount it faster upon the secondary or tertiary, quaternary repeated exposure to the pathogen. Does that make sense so far? So before we jump onto specifically humoral responses, we need to encounter and describe features of the antigen and antibody. Okay, I'm going to be relatively brief, and there are certain concepts that I don't touch upon here in details. We do it in the micro. So basically, for something to be an antigen, three conditions have to be met. The molecule has to be foreign, it has to be large, it has to be complex. Examples can include molecules such as proteins, carbohydrates, complex carbohydrates. I'm not talking about glucose, obviously, or DNA, or RNA for that matter. Now, let's illustrate each of these steps. So foreign. Let's say a human protein, a protein from, I don't know, Michaela's blood, like, I don't know, albumin, will not be foreign for me because we belong to the same species. When we say foreign, we refer to species, okay? The farther the species are from each other, the more quote-unquote foreign antigens are. So for us, something that comes from a mouse will be, well, obviously foreign, but probably something that comes from a bacteria will be even more unusual. Large, well, doesn't require an illustration. Complex, chemical complexity. So for example, materials like polyethylene that make up those bottles. Polyethylene molecules are really large, they're massive. But those polymers lack complexity. This is why our immune system cannot recognize them. And on the opposite end of it, are molecules that are relatively small but complex, such as many organic molecules. Basically, many of your hypersensitivities, many of your allergic responses, not so much, but let's say many responses such as poison ivy response, is the response triggered by a small molecule, which is chemically complex enough, and due to certain reasons that we don't talk about here, it suddenly manages to enlarge and trigger the immune response. But basically what I'm saying is, normally small, despite complex, but small molecules cannot stimulate your immune responses. Does that make sense? Like, such as hormones, for example. Good? Now, antigen is recognized by antibodies. So what is an antibody? Antibody is a protein, strictly a protein, which consists of two chains. So purple is the heavy chain, while red is the light chain. Chains are chemically connected to each other. It's not particularly important. What is important to understand is that antibody has a shape of a fork, right? You can see that. And as any proper fork, it has two prongs here, and it has a handle here. So prongs of a fork are called FAB fragments. So there are two FAB fragments in an antibody. AB stands for antigen binding. This part of the antibody, the handle, is called FC, or constant. Now, to some extent, we can illustrate it this way. Listen. When an antibody, look, with a fork, binds to an antigen, so with the prongs, binds to an antigen, the handle can then be recognized by multiple cells of your immune system, and the cells essentially can clear up the pathogen and just get rid of it. Does that make sense?
So does that attach to
like this?
Injuries that we have?
Well, not injuries, but it can attach to pathogens. So it specifically recognizes foreign objects, foreign molecules. Does that make sense? So if you have a torn muscle, nothing foreign is going on there. It's just torn, it's just physically damaged. So you're going to have local inflammation. You're not going to have accumulation of antibodies or T cells. Did I answer your question? Now, in terms of the antigen recognition, what's important to understand is that antibody doesn't just bind to the antigen. It binds to specific portions of it. So it would look something like this. So you have, and remember, it's all schematic, right? You have an antigen. Each antigen has multiple antigenic determinants. So this square, this circle, this little triangle, they represent antigenic determinants. What are those? Certain structures or sequences of, let's say, amino acids. Does that make sense? And if we will try to picture how antibody binds to it, it would look something like this. Okay? Take a look. So you can see that this antibody right here, right? It binds to the antigenic determinant of the antigen using its FAB fragment. I have to tell you, this is one of the most confusing for students to kind of process this interaction and formulate it, that FAB, antigen binding fragment of an antibody, binds to the antigenic determinant on the antigen. You have to process it, you have to simply memorize it, whatever, but this is one confusing part. Now, you may ask me, are antibodies that bind to this antigenic determinant or to this antigenic determinant, are they different from that one? The answer is absolutely yes. Yes. So each antigenic determinant is bound by a unique set of antibodies. Does that make sense?
When you mean unique, you mean just different?
They structurally different. Like, if you would look at the amino acid sequence, they will be different, okay? Now, I want just to say a few words. So, how many different types of antibodies can human body produce? The basic answer is unlimited number. Okay? Are you following me? Does that make sense? So we're talking about trillions and trillions of different antibody types. And at the very basic level, this is what happens. our genome contain several genomic fragments. Not for this exam. I promise there will be not a question about this, but I feel obliged to say a few words about it. So, our genome contains several genomic fragments. Now listen up. That encode different parts of the antibody. Does that make sense? And say you have 40 fragments that encode this. Okay? 5 fragments that encode this part and 6 that encode this part. By combining different fragments, it's called VDJ recombination. By this recombination, our immune system can create virtually unlimited specificity. You see what I'm trying to tell you? Think of it, to some extent, it's not a great analogy. It's a poor analogy, but to some extent it explains. Imagine that you have, okay, it's like a password. Okay? When you create a password and you have to include letters and numbers, if you randomly combine them, how many combinations you can produce? A lot, right? Does that make sense what I'm trying to tell you? So same goes here. Not only this, there are some other mechanisms that additionally increase diversity, but this VDJ recombination, the genomic recombination, creates a lot of breadth of antibody responses. We good? Okay.
You said that's not unattached, right?
It is not going to be on this exam, I promise that. Okay? And if I fail your expectations, you can take it to me and I will correct myself. Okay, we're good?